First of all, welcome everyone. Good morning, afternoon, or evening, depending on where you are in the world. Thanks for joining us again for another of our lectures in this series. It gives me great pleasure to introduce our speaker for today, who is Professor Lin Ye from Nanjing University. And Lin is an associate professor in the Environmental Engineering Department in the School of Environmental Science and Engineering at Nanjing University. And he did his bachelor's degree at Jianan University and a master's degree at Nanjing University. And then he went to Hong Kong and did his PhD at the Hong Kong University, which he finished in 2012. He then was a postdoc at IIES and Hong Kong University for 3 years before he joined Nanjing University in 2015. Dr. Ye's research mainly focuses on environmental biotechnology and microbial ecology. And he has an emphasis on the study of microbial diversity and function in wastewater treatment processes. And that's what he's going to talk to us about today. And so with that, I will turn it over to Professor Ye and welcome you and thank you for giving us your lecture.
Okay, Professor Ewan, thanks for your introduction. Okay. Oh, let me try to share my screen. Yeah. Can you see my screen?
Yes, that's good. Thank you.
Okay. Okay, this is Lin Ye from Nanjing University. I think without further delay, let's get started. And today my topic is understanding the biological wastewater treatment process with Metagenomics approach. So first of all, let me briefly introduce the biological wastewater treatment process. I guess maybe all of you have already know that about the activity slide process. This diagram shows the very traditional activity slide process, which was invented, I think, more than 100 years ago. But now it is still widely used in many wastewater treatment plants, especially in some municipal wastewater treatment plants. The process is very simple. The wastewater goes into the aeration tank where the microorganisms, also called activity sludge, remove the pollutants. Then the activity sludge and the treated water are separated in the clarifier. Besides the traditional activity sludge process, we also have many other biological wastewater treatment processes, such as SBR, MBR, USB, UBF, and so on. And there are many different kind of technology and different kind of processes. However, for all of these processes, the core and the key component is the microorganism. Therefore, to improve the biological wastewater treatments, we must understand the diversity and the functions of the microbial community. For example, if we want to improve the performance of the pollutants removal or want to reduce the energy consumption of this process, we must understand the mechanism of the microorganism in the bioreactor. Although the actual slide has been used for more than 100 years, the microbial diversity and the functions in these different wastewater treatment reactors is still not very clear because the community is very complicated and the tens of thousands of bacteria are present in the bioreactor or or other biological treatment system. So the community is very difficult and it's very complicated and very difficult to be studied. So now you may want to know why we need to use metaomics approaches to study the microorganisms in the wastewater treatment processes. Can we isolate them and study them one by one? Or can we study them by microscope? I think that the current answer is no. And I don't mean that we cannot use this method. We can use this method, and sometimes they are useful. But for most cases, they are not powerful enough because of the special characters of the microorganism. First of all, the microorganism is very small. For example, the size of the bacteria is only about 1 micrometer. And also they are very diverse. It is estimated that that there are 10 to 100,000 bacterial species on our Earth. And in the wastewater treatment bioreactor, usually there may be 1 to several thousand different species. Therefore, we cannot use microscope because under a microscope they look similar, and we cannot distinguish them. Moreover, many of these bacteria cannot be isolated in the lab. That means we cannot get pure culture. And therefore, it is difficult to study them with the traditional microbiological method. Therefore, we need to use some other method to study the microorganism. It is well known that all functions of the microorganisms are determined by the DNA of the microorganism, and the proteins are synthesized based on the information on the DNA through the transcription and the translation. And then the proteins are used to catalyze the pollutant degradation. And therefore, based on this, we can study the functions of the microorganism in the wastewater treatment bioreactor based on the investigating of these macromolecules, DNA, RNA, and protein. So we have these metaomics approaches, the metagenomics, metatranscriptomics, and the MetaProteomics. And these metaomics approaches are used to study DNA, RNA, and protein respectively. Here, I would like to explain a little bit about the difference between genomics and metagenomics. Metagenomics is used to study the genomic material recovered directly from the environmental sample, while genomics usually means the study of the genetic material of one organism, or pure culture bacteria, pure culture archaea. They are different. The same rule also applies to the transcriptomics and proteomics. For wastewater treatment, we usually use a complex microbial community instead of a single microorganism. For all wastewater treatment bioreactor, I think no one use a single bacteria, single microorganism to treat the wastewater. Almost all for all bioreactors, they are very complex microbial community in the bioreactor. Therefore, the metaomics is usually used for analyzing the microbial community in wastewater treatment bioreactor. Besides, we also have some other omics approaches, such as metabolomics, lipidomics, but these methods currently are not widely used in the environmental microbiology. Okay, now let me introduce these three metaomics approaches one by one. As mentioned above, metagenomics is the study of a genetic material, also called a metagenome, recovered directly from the environmental sample. By using this method, we can obtain information on the microbial diversity functions without isolation and lab cultivation with individual species. We just need to extract DNA of the sample and do the sequencing. And after that, we need to do some data analysis. Then we get the microbial diversity and the function information from the sample. This is particularly useful for studying the mechanism of the biological wastewater treatment process. Because when we investigate a wastewater treatment reactor, we always want to know the diversity of microorganisms and their function in the reactor. Here is an example. A few years ago, maybe I think in 2010 or 2011, we investigated the bacterial community in the active sludge from several different municipal wastewater treatment plants in different locations of the world. Some from mainland China, and some from Hong Kong, Singapore, and some from North America. Before that, the overall bacterial diversity in the active sludge is not very clear. Although it had been used, the active sludge process had been used to treat wastewater for more than 100 years. The diversity of the microbial diversity of active sludge is not very clear. In this study, we found that the bacterial community was very diverse in the active sludge, and also we found a core population among the sludge from different locations. However, we also found that some bacteria, the abundance of some bacterial species that are largely different from different wastewater treatment plant. And more interestingly, we found that a clear geographic difference among the active sludge sampled from Asia and from North America. As you can see from this figure, the sample from Asia and from North America are clearly separated. This is probably due to the different wastewater composition or different plant design or operational conditions in different plants. And besides the community structure, the metagenomics approach can also be used to study the specific gene. This is an example that uses metagenomics to study the genes of Anammox bacteria. I'm not sure whether you are familiar with the Anammox bacteria. They are involved in wastewater treatment process to remove nitrogen from wastewater. And they also present in other environments such as soil, rainwater, and the soil. The Anammox process had many advantages. For example, it does not require aeration, and the less sludge produced in the process. However, none of Anammox bacteria had been obtained at the pure culture. So far, there are no pure cultural Anammox bacteria. Therefore, the metagenomics is a key measure to access their genes and get a better understanding of the bacteria in different bioreactors. And also we can use a metagenomics method to recover genome to study the function of a specific microorganism. This study presented a very useful approach to recover high quality microbial genome from the metagenomic sequencing. And for example, for Anammox bacteria, currently there is no pure culture, so we cannot we can use this method to get the draft genome from the environmental sample and then to analyze their function. For the all bacteria, if they cannot be isolated, a researcher can obtain their genome by using this method. And metagenomics tells us which microorganisms are present and what genome potential or function they have. While sometimes we also need to know the activity of the microorganism. So metatranscriptomics can tell us about their activity. First, we need to know what is the metatranscriptome. Metatranscriptome is the total content of the gene transcript in a community at a specific moment of sampling. The metatranscriptome is not a constant. It varies with time and the environmental condition. So then what is the metatranscriptomics? Metatranscriptomics are the techniques applied to obtain the whole expression profile in a community and to follow the dynamics of gene expression pattern over time and the environmental pattern. By using metatranscriptome, we can answer the following two questions. The first one is which are the highest expressed genes in a specific environment? And the second is which gene should the highest change in expression level between different conditions. The first one is for studying the single environment, and the second one is for comparison. We can compare the expression level between different conditions or different samples. This study used the metatranscriptomics to study the co-expression pattern of polyphosphate accumulating organism during the enhanced biological phosphor removal cycle. Polyphosphate accumulating organism, also called PAO, are a group of very important bacteria and for phosphate removal under anaerobic and aerobic conditions in the wastewater treatment reactor. They can release phosphate under anaerobic condition and absorb phosphate under aerobic condition. So by using metatranscriptomic approach, we can know which genes were overexpressed under anaerobic conditions and which were overexpressed under aerobic conditions. Based on this information, the metabolism of the polyphosphate accumulating organism can be explored. Okay, then the third one is MetaProteomics. MetaProteomics is the study of all protein recovered directly from the environmental sample. So far, the protein sequencing is more difficult than DNA sequencing. So the MetaProteomics is not that widely used, although it is very important. For metaproteomics analysis, first we need to separate the protein on a geo. Usually the two-dimensional page or the SH page geo electrophoresis technology are used. these are two different electrophoresis technology, and the protein can be separated according to their isoelectric point and mass. Then the mass spectrometric is used for the protein identification. And also in recent years, some gel-free methods have also been developed to separate the protein. And this study used MetaProteomics to study the active sludge in pharmaceutical wastewater treatment reactor, and identified the most constantly expressed protein and the corresponding bacterial species. Due to the time limitation, I will not talk too much about this. Okay, in the following slide, I will present some detailed information about the metagenomics method, because it is the most widely used method at present. Before talking about the metagenomics, I would like to mention a little bit about the high throughput sequencing, which is also called next generation sequencing. The high throughput sequencing is a revolutionary DNA sequencing technology invented a few years ago, and it significantly changed the study of biology. It can parallelize the sequencing process and generate a huge amount of DNA sequences concurrently at a very low cost. The cost is extremely low compared with the traditional sequencing method. So it is very widely used now. And to take this machine as an example, it is a sequencer produced by Illumina. This is a very big company. And this machine can generate 400 gigabase pair in 1 day. That's a very huge data set. I think almost equal to the whole volume of the hard disk in my computer. Therefore, the data analysis of metagenomic sequencing cannot be done in our personal computer, and usually we need to use some very powerful computer, and for the memory and the hard disk and the CPU, the requirement is very high for the high throughput sequencing data analysis. And based on this technology, the metagenomics, based on the high throughput sequencing technology, the metagenomics had been developed very fast in recent years, and had been widely used in the research of biology, including the environmental microbiology. So to investigate a biological wastewater treatment process or bioreactor, we are always care about two questions. The first one is which microorganisms are present in the reactor, and the second one is what are they doing? What function do they have? So to answer these two questions, we can use 16S RNA gene sequencing and the shotgun metagenomic sequencing respectively. The shotgun metagenomic sequencing is also called metagenomic sequencing in some cases. First let's look at the 16S RNA gene sequencing. I'm not sure whether you are familiar with the 16 RNA gene. It's a gene only found in prokaryote, and it codes for the small subunit of ribosome. And more importantly, it is commonly used genetic marker for identification and classification of bacteria and archaea. This is very important. The 16S RNA gene sequencing is a widely used technique for identification and classification of bacteria and archaea in environmental sample. And also this is a very basic technology and had been used many years. I mean, not only for the high throughput sequencing. Before this, in the previously we also used the traditional sequencing to sequence the 16S RNG, and this figure. This figure shows that the 16S RNG has some controlled regions and variable regions. For the controlled region, that means these regions for different bacteria, these regions are similar. The base pair in these regions are similar. Therefore, the controlled region can allow us to design PCR primer to do the amplification. And this diagram shows the general protocol of 16 RNG high throughput sequencing. Suppose we have several different activity site samples, and would like to analyze the bacterial community in each sample. First, we need to extract the DNA from this sample, and then use PCR to amplify the 16S RNG. Please note that the PCR primer used for the different sample has different tags. Sometimes they also called barcode, which can be used to distinguish the sequence from different samples. Then the PCR products were mixed and sequenced on the high throughput sequencing machine. After that, we need to do some bioinformatic analysis on the sequences and to explore the bacterial community in this sample. For the bioinformatic analysis, I think it is the most hard part of the metagenomics study. Sometimes it's very time consuming. Here I listed 3 most commonly used software for 16S RNG sequencing data analysis and their website. If you are interested in the data analysis, you can check the website and read the material. And usually on the website there should be example data and the tutorial. You can download the example data and go through the tutorial, then the procedure of the data analysis. And besides of the community structure, sometimes we also need to know the detailed information of the microbial community. Therefore, we need to sequence all the DNA in the sample instead of a specific region. That is a shotgun metagenomics sequencing. The procedure is very simple. We just need to extract DNA and do some treatment on the DNA, and then we can do the sequencing and get the data. Usually, in most labs, you just need to extract the DNA, then send the DNA sample to a commercial company. Then after a few days, you will get the data. But there's a problem. Currently, the sequences we get are usually very short. For example, the sequence length of Illumina sequencer is only about 115 to 300 base pair. That's very short. Although some other companies have developed some machines that can generate long sequences, but their error rate is usually very high. Therefore, they haven't been widely used. Due to the short sequence length, sometimes we need to do assembly first to get a longer sequence before conducting further analysis. And for the metagenomic data analysis, there are many different methods. One method is to directly compare the sequence data with a specific database. By using this method, we can explore some known genes, such as genes involved in nitrogen removal, aromatics removal, and we can, sometimes we can also explore the antibiotics resistant genes. And then the diversity and the relative abundance of these genes can, from different samples, can be compared. But if you use this method, you must have a database. If you want to investigate some genes, but you don't have a database, you cannot use method. Before you did the method, you must have a database, or you build a database yourself, and moreover, due to the short read length, sometimes the comparison result may be not very reliable because the sequence length is too short. The result could be, the comparison result could be not reliable, and and moreover, by using this method, we cannot know the host of the gene. We can only know the function of a gene, but we cannot know which bacteria carries this gene. And another method is that we can use assembly method, where we can first assemble the short read into long contigard. The long contigard means longer sequences. As shown in this figure, the long contigard can be obtained through the assembly, and then the contigard can be classified based on the coverage and the chemical profile. And then and based on this information, the contigard can be grouped into different beans. We call this process binning, and that means we can recover draft genome from the metagenomic sequence data. Then we can analyze the taxonomy and the functions of each genome. In this process, the binning is a very important step. For the genomic binning, there are a few software such as Concact, MetaBet, Genome, Group M, and so on. There are also many other binning software. If you are interested in the genome binning, please check their paper or website. And for the metagenomics analysis, another important step is the annotation. Through the annotation, we can know the function of different genes. Here I listed a few database and software for the metagenome annotation. Except the database and the software, there are also some online analysis system. For this system, you just need to upload the data. Then they can be analyzed automatically. Then after a few days, maybe sometime after a few months, they will send the result to you. And here is only a very brief list. There are also many other good databases that can be used. Okay, that's all. Thank you.
Okay, thank you very much, Dr. Yu. Are there questions for our speaker today? Any questions for our speaker? I see you've got one question here in the chat, Lynn.
Okay. Can you clarify what the term binning based on quantum. Let me this for this slide, the opinion. So after assembly, we get a lot of long sequences. So we get then we need to use some measure to know which sequences are from one bacteria and which sequences are from another bacteria. There are a lot of information we can use. For example, we can map the row read to the contigard, and if the three contigard, if some of the contigard are from one bacteria, the coverage should be same. For example, if one bacteria account for 10%, and another bacteria account for 20% in the original sample, and after sequencing, the coverage should be different, and the coverage of the bacteria with higher burden should be larger than the other bacteria. Based on this information, we can group, and also we can calculate the chimer profile. What the chimer means a few of base pair. For example, we can the key can be 6, 4, or 10. We can calculate the chimer profile to group the contigard, and we also can calculate the GC value. For some bacteria, they have low GC value, and for some bacteria, they have a high GC value. Based on this information, we can group the contigard into different groups, and this process is binning. After binning, we can analyze each bin individually. Okay. Is that clear? Pause. Regarding the microorganism difference in wastewater treatment from different regions, we listed the composition as one region. Do you know which microorganism in high proportion of pesticide? Okay. Okay. For this question. The study we conducted is for the municipal wastewater treatment plant. For most municipal wastewater treatment, there are no pesticide. So we didn't investigate the pesticide. Actually, now we are doing a project. We would like to investigate some emerging pollutants, and including the pesticide, herbicide, and maybe antibiotics and other pollutants. Maybe sometime later we can have answer for this question. Any other question? how come the usual water treatment? How common the usual actually in the field of I think for most study, if they want to investigate the microbial community, I think more there should be more than 90%. 90% should use the hash precipitation. This is a very commonly used method. I think before 2010, people use DGGE or TIFLP or some other method. But currently, most people use the hash precipitation, and almost no people use the other method. For example, the DGGE, TIFLP, and also some people use FIS, FISH. It's a in situ method. But it's the experimental procedure is very complicated, and also the resolution is not very high, and that's the reason I think that's why hash precipitation is widely used.
Do we have any further questions for our speaker? So I'll give one last chance here. Otherwise, I want to thank you, Lynn, for a very interesting talk. Very helpful, puts into perspective some of the things that we have heard in some of our earlier lectures. I just want to remind everyone that Hui Dang will stay on the line, and there will be a discussion of the lecture today for anyone who is interested in taking part in that discussion. And to let you know that our speaker next week will be talking on the SANI process, the use of sulfur in waste treatment. And our speaker is from Sun Yat-sen University next week. So with that, I'd like to thank you again, Professor Yu, for your lecture today, and thank everyone who has been listening in. See you next week.
See you.